Introduction to Food Microbiology. Introduction to the module. The study of microbes that inhabit, create, or contaminate food is known as food microbiology. Microbes are present in the water we drink, the food we consume, and the air we breathe. Food microbiology is a vast field that includes the study of microorganisms that have both good and detrimental effects on the quality and safety of raw and processed foods. This module will give you a basic insight into the food microbiology and major sources of microbes. Learning objectives. After finishing this module, you will be able to define the concept of food microbiology, understand the history and current scenario of food microbiology, point out the sources of microorganisms. What is food microbiology? The study of microorganisms that inhabit, create, or contaminate food is known as food microbiology, including the investigation of microorganisms that cause food spoilage. However, beneficial bacteria, like probiotics, are becoming increasingly significant in food science. Furthermore, microorganisms are required to produce fermented dairy products and bread, beer, and wine. Many microorganisms are found naturally in foods, sometimes as contaminants. Bacteria are the most prominent, both as pathogens and as spoilage microorganisms. Even though these invisible foes are tiny, they have the power to kill species that are many times bigger and heavier than themselves. The bacterial and fungal attack causes between 1/4 and 1/2 of the world's food supply to be lost after harvest. Although microbiological food spoilage may not result in any deaths or illnesses, it costs industries and consumers millions of pounds per year. It also constitutes significant resource waste, which is the ugly side of microbial existence. Therefore, in order to comprehend and effectively address the microbial issues related to food, current food microbiology requires a significant amount of basic science. The field involves fundamental knowledge of microbial ecology, physiology, metabolism, and genetics. History of food microbiology. It is reasonable to assume that our ancestors, the hunters and gatherers, were aware of food deterioration and foodborne infections. Even without knowledge of the causative components, people used ice and fire to preserve and protect foods. Some food preservation methods are drying, cooking, baking, smoking, salting, sugaring with honey, spicing. These methods were used between 8000 and 1000 BC, most likely to reduce spoilage. Many societies widely employed fermentation not just to preserve goods but also to make a variety of attractive foods from milk, meat, fish, eggs, grains, fruits, and vegetables. Following Leeuwenhoek's discovery of the ubiquitous existence of microorganisms, primarily bacteria and yeasts, in the 1670s, some people began associating these organisms with food spoilage, fermentation, and foodborne diseases. Pasteur pioneered the major developments of thoughts on the possible roles of microbes in foods and their scientific proof in the 1870s, and many other scientists followed suit before the end of the 19th century. This cleared the path for the early creation of food microbiology in the 20th century. Current scenario of food microbiology. Studies on the relationship and significance of microorganisms, particularly harmful bacteria in food, began throughout the early 20th century. Their isolation and identification required the development of specific techniques. It was acknowledged how crucial sanitation was in handling food to prevent microbial contamination. Specific techniques were investigated for both growth inhibition and pathogenic and bacterial spoilage destruction. Additionally, there was some interest in identifying helpful microorganisms linked to food fermentation, dairy fermentation, researching their individual traits. Understanding the microbial population and microbial interaction in a complex food environment is now at the forefront of food microbiology due to microbiome analysis, whole genome sequencing, and the advent of novel and natural intervention tools. Sources of microbes in food. Microorganisms enter foods through both natural and external sources in which food comes into contact from the time it is produced until it is consumed. Understanding the sources of microorganisms in food is essential for creating methods to control access of some microorganisms to food, developing processing methods to kill them in food, Determining the microbiological quality of food. Establishing microbiological standards and specifications of foods and food ingredients. The main sources of microbes in food are discussed below. Air. Dust and moisture droplets in the air both contain microorganisms. They are temporary and variable, dependent on the surroundings rather than growing in the dust. Air can contain spores of Bacillus spp, Clostridium spp, Micrococcus spp, Sarcina spp. Different species of bacteria, including pathogens and viruses, can be spread through the air if the environment contains a source of pathogens, for example, livestock and poultry farms or sewage treatment plants. It is possible to reduce microbial contamination of food from the air by eliminating the probable sources, regulating dust particles in the air, using filtered air, utilizing positive air pressure, lowering humidity levels, adding UV lighting. Soil. There are many different types of microorganisms in the soil, notably soil used to grow agricultural products and nurture animals and birds. Because microbes can reproduce in soil, they can be found in large quantities. The soil can harbor a wide variety of molds, yeasts, and bacteria, including Enterobacter, Pseudomonas, Micrococcus, Enterococcus, Bacillus, Clostridium. Various parasite species can also enter food through the soil. Removing dirt from foods by washing them and avoiding soil contamination can minimize the number of germs in certain items. Water. Food is produced, processed, and, under certain circumstances, stored using water. It is used to irrigate crops, provide drinking water for farm animals, raise fish and marine goods, wash and prepare food, store food and clean equipment, operate processing and transportation facilities. Numerous processed meals also contain water as an ingredient. Although drinkable water is free of coliforms and pathogens, it may include other bacteria that cause food spoiling, such as Pseudomonas, alkaligenes, Flavobacterium. Water that has not been properly treated may include dangerous and spoilage microbes. Humans. Foods come into contact with many people, handling them between manufacture and consumption. They include persons who work on farms and in food processing factories, as well as those who handle food in restaurants, catering services, retail stores, and at home. Human carriers have been identified as the source of pathogenic microbes in meals that have subsequently caused foodborne infections, particularly in ready-to-eat foods. Substantial human causes of microbial contamination in food include unclean hands, lack of aesthetic sense and personal cleanliness, unclean clothes and hair. To limit contamination from this source, proper training of staff in personal hygiene, regular health checks, and the maintenance of efficient sanitary and aesthetic standards are required. Highlights from this module. In this module, we have covered the following points. Bacteria are the most prominent, both as pathogens and as spoilage microorganisms. Dust and moisture droplets in the air both contain microorganisms. Numerous processed meals also contain water as an ingredient. Microbes can reproduce in soil, so they can be found in large quantities in the soil. Well done. You have reached the end of this module.